In this video, we are going to talk about endocytosis, its types, pinocytosis, and phagocytosis, where they are taken up, from where do we get the energy, and how these structures are formed. So, endocytosis is divided into pinocytosis and phagocytosis. The cells require nutrients. So, from where do they get these? They get these by simple diffusion and active transport. Simple diffusion is the movement from high concentration to low concentration. And in active transport, we require ATP that is formed in the mitochondria. Few of the substances go through the pores, and for example, lipid soluble substances like CO2 and O2, they go through the lipid matrix. Larger substances require the process called endocytosis. So, in pinocytosis, ingestion of minute particles that form the vesicles of the ECF and particulate components into the cytoplasm. It usually occurs in the cell, like continuously happening. The size is 100 to 200 nanometers, and mostly protein goes in through this method. How about the size? The size can be variable. For example, in case of macrophages, within a minute 3% of macrophages will be engulfed. So it is variable or dependent. We should know the exact process of the pinocytosis. Before that, let's know a little bit about the structure. Here we have the coated bits outside the membrane, and inside the membrane we have the clathrin. These are fibrillar proteins. Now here are the proteins which are coming through this method. So these proteins attaches to these special receptors. Here are the special receptors. Now these receptors and attachment of protein will cause the surface properties to change. How these are changed, we are unsure of that, but we know that there will be requirement of ATP or the high energy source, and we will require calcium from the ESF. Why calcium is required? Calcium is required to attach with the fibrillar protein, that is clathrin. Now, because of these changes, the entire pit invaginates downwards or inside the cell. Now the borders will close and it breaks away from the cell. These breaking away from the cell, there is a vesicle formed, a circle type of organelle is formed. That is called the formation of pinocytotic vesicle. The second is phagocytosis. In case of phagocytosis, there is ingestion of large particles. Large particles like we have bacteria, the whole cell, or even the degenerative tissue. There is no difference from the pinocytosis. The difference only is that they engulf the large particles. So, for example, who does it? It is done by macrophages and some of the WBCs. Like suggested before in pinocytosis, that they require the attachment to the special receptors. Here also, it is initiated when they bind with the receptors on the surface of the phagocyte. But especially in case of bacteria, they are already attached with the antibody. So antibody here will attach to these specific receptors. So here is the attachment of antibody, and then here will be the bacteria. So because of the attachment of antibody to the special receptors, it will bring the bacteria to itself, and this process is called opsonization, which we will do in the later videos. So in general, the difference is, for example, if you're being naughty and you're playing with mud and toys, your mother comes in and clean up the mud that you have spilled over. So this is the small particles, that is pinocytosis, and your father comes in and cleans up the toys you have been playing with. That is phagocytosis, because toys are the large particles. After this, what happens? They should be taken away. They should be digested away. So that is, we have known the lysosomes are the intracellular digestive enzymes. The lysosomes comes in and take the pinocytotic or phagocytotic vesicles, in which there is a formation of digestive vesicle. For example, you eat some food is digested, and let's say 20% of them is undigested. So these undigested part here will be termed as residual bodies, and these residual body will come out or excreted. For example, you eat, yeah, some part is digested, and then you go to the toilet. that is called exocytosis. We should know other roles of the lysosomes as well. There can be removal of damaged cell. Damaged cell can be severe or slightly damaged. In case it is slightly damaged, then we remove half of the cell or the part of the cell. If it is very severe damage, then the whole cell is taken away. So this is called autolysis. Other roles of the lysosome is the removal of regressive tissue. So for example, we have uterus after pregnancy, we have mammary glands after lactation, and we can have muscles also after a long period of inactivity. In lysosomes, these are the hydrolases present. These hydrolases are activated when the acidic pH is present, that is, the acidic pH is 5. Other agents here present will be lysozymes and lysoferrin. Lysoferrin will bind to the iron, which promotes the cell growth, and lysozyme dissolves the bacterial cell. Next, here we heard about the term the autolysis. Autolysis or autophagy. Autophagy means to eat one cell. The worn out cell come out in the form of double membrane. Here is the double membrane. These when these are enclosed in the double membrane, these will be termed as autophagosome. Now, autophagosome are also taken away from the by the help of lysozymes. Here hydrolyses are present, and these will lead to the breakdown and degradation. Why do we need such process in our body? It is required for the key mechanism in tissue development, and it is required for the maintenance of homeostasis. These are required when nutrients are less, so for the cell survival. Now, from where does the formation of structures happen? Like we have already discussed, it happens in the endoplasmic reticulum, that is, rough endoplasmic reticulum and smooth endoplasmic reticulum, and the Golgi apparatus. In the rough endoplasmic reticulum, there is a formation of protein. In smooth endoplasmic reticulum, there is a formation of lipid or the synthesis of lipid. In the Golgi apparatus, I have mentioned about chondroitin sulfate and hyaluronic acid. These are the major component of proteoglycans that is secreted in the mucus or in the other glandular parts, or these are the major component of ground substance, or these are non-fibrous component of interstitial space. These can be also the component of cartilage and bone. These are also required for the cell proliferation and migration. For example, when the protein is coming out from the Golgi apparatus in the form of secretory vesicles, first the secretory vesicles is diffused and it is fused in the cell membrane. Now, after this, the proteins are released by the help of exocytosis, and these secretory vesicles are attached to their own organelles for the growth, or they are taken away by the lysosomes. Now, from where do we get the energy? Like how the energy comes to us to have such processes in the cell? It is because of the mitochondria. How does the mitochondria helps? From how do the mitochondria gets the energy itself? We are eating. So we are eating carbs, proteins, and fats. Carbs will turn into glucose, proteins will turn into amino acids, and fat will turn into fatty acid. These are oxidative reactions. In these oxidative reactions, there is release of energy inside the mitochondria, and as we know, for the formation of ATP, we need energy. So, how exactly in the mitochondria? First of all, the glucose formed from the carbs will turn into pyruvic acid. Here also, like I said, there is oxidative reactions going on. So, the ADP, sorry, will turn into ATP. So, here is the formation of ATP. Now, after this, the pyruvic acid will go into the matrix. This is the matrix of the mitochondria, and it will lead to the formation of acetyl coenzyme A. The acetyl coenzyme A will further go into Krebs cycle or citric acid cycle, CAC. In the citric acid cycle specifically, the acetyl coenzyme A releases hydrogen atoms and carbon dioxide. Carbon dioxide will diffuse out, and hydrogen will combine with oxygen to form water. To form water, we need ATP synthetase, that is, the protein enzyme present in the membrane of the mitochondria. This will again lead to the release of energy, and the ATP formation takes place. Now these new ATP goes out of the mitochondria and utilized where it is necessary. Some points about ATP is that it is a nucleotide, that is three parts. It is of three parts. The nitrogenous base, that is adenine. Then we have the pentose sugar, that is the ribose. Then we have three phosphate radicals, that is, here are the three phosphate radicals. It is a high energy bond and splits off easily. These bonds on the second, the last two bonds of the phosphate will will attach to the other molecule of the ATP. Now, when the ATP is released, there is again release of energy, and there will be release of phosphoric acid and radical shift, and the radicals split away. This leads to the formation of ADP. Now, again, from somewhere we will have the release of energy. These ATP can bind with the phosphoric acid, which leads to the formation of ATP. So that's all in this video.